Dear students, once again I am meeting you, discuss more about polymerase chain reaction. The last class I discussed more the events what happened during polymerase chain reaction with the diagrams. And here also a simple diagram in a nutshell showing the polymerase chain reaction. Now we are taking what is called the targeted sequence of DNA, the gene of interest. Now we are adding, so we are taking a mixture in the form of what is called this DNA sequence, then tag polymerase, and also the primer, the nucleotides, etc. What we are doing normally, heating, cooling, that is called annealing, adding primers to get single stranded DNA, that primers anneal to. The meaning for that word, that primers anneal to, hybridize with the template stranded DNA. Now, the reaction requires, so the two strands get separated. These are the two single stranded template DNA. Now we are adding this tag polymerase for the extension process. For that it needs energy for the extension of DNA. The energy source may be in the form of DATP, deoxyadenosine triphosphate, or DGTP, deoxyguanosine triphosphate, or DCTP, deoxycytidine triphosphate, or DTTP, thymidine triphosphate. These are all the energy which provide, actually energy sources, they provide energy for the process to synthesize the complementary strand. Now this one synthesizes its own complementary strand, this one synthesizes its own complementary strand. Likewise, the cycle being repeated for many times so that we are getting billions of copies. Here in the book is given the cycle is being repeated 25 to 75 times. Okay, given already this picture in the previous what's called video. You go through that one. The next one we have to go for PCR in what is called clinical diagnosis. What is the role of PCR in clinical diagnosis? Let's see now. Now, what is the role of PCR in clinical diagnosis? You know the PCR is highly specific and sensitive. So the specificity and sensitivity of PCR is useful for diagnosis of different diseases like inherited diseases such as genetic disorders, the bacterial diseases and viral diseases. And see that one, the diagnosis and treatment of a particular disease. If you want to treat a particular disease, you have to diagnose it. So, the diagnosis and treatment of a particular disease often requires, see that one particularly for identifying a particular pathogen involved in causing the disease. You have to identify the pathogen. So, the traditional methods or conventional method of identification involve just actually culturing these organisms from a clinical specimen. Clinical specimen means the blood sample, or we have the urine sample, or the sputum sample, or the spinal fluid, or the stool. Anything which is collected for test is called as a clinical specimen. Okay, that involves, you see that one, collecting these actually the materials from clinical specimen and culturing it, and then performing different tests, metabolic and other tests to identify the specimen. This is what we call the identification of the disease causing pathogens by traditional methods. So we have to culture and we have to do the test, either metabolic test or any other biochemical test to identify. But the concept behind PCR based diagnosis of disease, the concept behind PCR-based diagnosis of disease is very simple. Suppose in a particular specimen, what's called clinical specimen, either blood or stool or sputum, if pathogen is present in the particular specimen, it means that its DNA will also be present in that sample. So we are doing the molecular analysis. If pathogen is present in a clinical specimen, that is, its DNA will also be present. So we can normally just detect the DNA and we can just do the test by PCR from these samples and identify what we have actually the type of antigen and the type of pathogen and the earliest clinical diagnosis. Okay, now what are the uses of this one and what are the different clinical applications we can see? One application from the clinical specimen, we are identifying the DNA. If pathogen is present, its DNA is also present. Let's see. By using the DNA, how can you identify, how can you detect a particular pathogen causing the disease? Now we see that when the pathogen's DNA has a specific sequence, the one which is present in the clinical specimen, that can be detected by using what's called PCR. By often using some clinical specimen in the form of, you see, blood, stool, sputum or spinal fluid, etc. in the PCR mixture. So, if pathogen is present in a mixture or in a clinical specimen, it means it also has what we call the DNA. Now, the pathogen DNA has a specific sequence. You know that each and every organism has a specific sequence of DNA. That specific sequence can be detected by PCR in a clinical specimen like blood, stool, sputum or spinal fluid in the PCR mixture. Another what we have usage in clinical diagnosis. Now PCR can also be employed in prenatal diagnosis before the birth. Prenatal means before the birth. So before the birth you see that one, you can find out any genetic diseases in the developing embryo. So that is called prenatal diagnosis. So PCR can also be employed in prenatal diagnosis of inherited diseases just like Down syndrome or any other syndrome, Pato syndrome, etc. By using what's called chorionic villi. The villi is nothing but finger-like projections formed by what is called the chorioallantois placenta of humans, which fits into the cavities or the crypts of what is called actually the uterine wall together form the placenta. So, by obtaining the cells from the chorionic villi samples, that is, it is the component of placenta, or cells of amniocentesis. Amniocentesis is a process by which the amniotic fluid is sucked out by using hypodermic syringe. To analyze the cells found in that what is called amniotic fluid to find out the gender or sex of the baby, because the cells found in the amniotic fluid also similar to the cells of the embryo. The process of sucking out the fluid from the abdominal, that is what is called the womb of the mother, that is from the amniotic cavity, that process called amniocentesis. So the cells of amniocentesis, after the process, after sucking the fluid, amniotic fluid, that is from what is called amniotic cavity, the cells have been collected and used for prenatal diagnosis before the birth of the child. In the developing embryo, is there any genetic diseases? For example, the diseases like sickle cell anemia, an autosomal recessive gene disorder, beta thalassemia, another autosomal recessive gene disorder, and phenylketonuria, another autosomal recessive gene disorder. These are all the diseases that can be detected in the case of developing embryo before birth by using what is called PCR. By using these samples, either what we have the chorionic villi samples or cells of amniocentesis. I mentioned once again, chorionic villi is nothing but a component of the placenta. Cells of amniocentesis, the process by which the amniotic fluid is sucked out and take the cells from that fluid. Then, another important just actually clinical diagnosis. Now we have cDNA, the complementary DNA that is being produced by means of reverse transcription process using reverse transcriptase enzyme, what I mentioned. That is what is called RT-PCR. So the cDNA from PCR is a valuable tool. What for? For diagnosis and for monitoring retroviral infections. Because you see that one, the process is carried out by using the reverse transcriptase enzyme. Reverse transcription process normally occurs in the case of what we have the retroviruses having RNA genome and that is being multiplied through DNA. So, to find out the retroviral infections for monitoring as well as for diagnosis, the cDNA that is used from PCR is normally find out any diseases or find out or to look out or to monitor the retroviral infections. Is there any retroviral infection? In the book it is given what is called a bacterial disease, tuberculosis. by what is called Mycobacterium tuberculosis. So it is not a retrovirus, it is a bacterium only, the one which causes what we have the disease. An example is given in the book. So anyway, the cDNA from PCR is useful for diagnosis and monitoring of retroviral infections caused mostly by the retroviruses like HIV, hepatitis B. These are all the retroviruses. Okay, right? Let's see furthermore about the clinical use of PCR. Several virally induced cancers, such as you see that one cervical cancer, cancer in the neck region, particularly the neck of, that is the uterus. The neck of uterus is called cervix. It is not the neck region here, it is the neck of uterus. So very common cancer in females. So cancer such as cervical cancer, that one is caused by papilloma virus, and that can be detected by PCR. So cervical cancer of the uterus can be detected by PCR. The next one, the sex of humans and also the livestock and also the embryos fertilized in vitro, that is outside the body, can be determined by using what's called a primer DNA or using DNA probe. DNA probe means the enquirer, the one which is used to enquire, the find out, specific for sex chromosomes. So we have sex of humans and living stock. Embryos fertilized in vitro can be determined by PCR using the primers, the DNA, and DNA probe, the enquirer, to find out. So DNA probe is nothing but a small segment of DNA which is used to identify the DNA of any other species, specific for sex chromosome. This DNA probe is specific for only sex chromosomes to find out the nature of the sex chromosome. So we have a number of sex-linked disorders. These sex-linked disorders, in the case of fertilized embryos, can also be detected by PCR. So sex-linked disorders in fertilized embryos can also be detected by PCR. So these are all some of the what is called actually the role of PCR in clinical diagnosis. Now we have to conclude of the PCR, the next one, applications of PCR. What are the applications of PCR in the present generation as well as for future? Now, the differences between the two genomes or difference between the genomes of two different organisms can be studied only through what is called PCR, by gene amplification or DNA amplification. Second one, it also plays an important role in the field of evolution or study evolution. More specifically, the phylogenetics. You know the phylogenetic system of classification. Based on the evolutionary principles, we can classify the organisms. Then, so it is a technique used for amplification of DNA. As a technique which can amplify even minute quantities of DNA from any source. For example, one hair. But the cell must have what is called a nucleus with the DNA. That's very important. Not a dead cells without DNA. So such as, for example, hair, mummified tissues, you know the mummies which lived, actually we have in Egypt, we have the mummies. They are actually preserved human beings with the help of herbal what is called smearings. So the mummified tissues, the persons who lived many years ago, bones or any fossilized materials. From these source, even a minute quantity of DNA is enough to amplify with the help of PCR. So I mentioned about only just you can do it in the case of dead cells, so in the case of living cells. And the meaning for that one, the cells which have a nucleus. The nucleus only contains a DNA. Now the RBCs cannot be used because it contains no what is called nucleus and so also DNA. So we have small amount of DNA is present in the hair or in the case of mummified tissues or bones or any fossilized material, by which only we can just actually find out the life on earth just before 200 million years or 100 million years ago, the organisms which lived, by DNA analysis using DNA amplification. Then what is the role of PCR? PCR plays an important role in what's called forensic science, particularly in making what's called DNA fingerprinting. Okay, so PCR technique can also be used in the field of forensic science. I will explain now. in forensic science, gene amplification or DNA amplification can be performed by what is called PCR. So in that one, a single molecule of DNA is enough from any source. For example, from the blood stain or from hair or from the semen of an individual is adequate for the amplification of, that is what is called DNA by PCR. One single DNA molecule can be multiplied to form billions of copies to identify what is called the criminals in forensic science. Now the amplified DNA is used, you know that one, to develop the DNA fingerprint. We have already seen DNA fingerprinting is an important tool. You see that one in the field of forensic science. How? Now this is used in the identification of criminals. So DNA fingerprinting is normally used to identify the criminals, and for DNA fingerprinting we need a single DNA molecule obtained from the criminal scene or any other source to be multiplied, and that is by means of what we have, amplification of DNA through PCR. So that's about the role of what is called PCR in forensic science for amplification of DNA that is used to develop what is called DNA fingerprint, which is an important tool in the forensic science. Thus we can identify the criminals. The last one, PCR can also be used to just amplify what we have, a specific DNA segment that can be used in gene therapy. Suppose a particular gene is defective, you know, in gene therapy we are replacing the defective gene with a corrective gene. That is what's called a specific DNA segment that is to be amplified. Just one gene is enough. Just one gene is enough. So just one gene is, I can say, enough to what is called the replace the defective genes, not sufficient. So just one gene is not enough to replace the defective genes. That's why we have to amplify the genes before doing the gene therapy. That amplification is performed by means of PCR. The particular gene of interest has to be amplified before it is being actually reimplanted in the body of what is called a patient having a defective gene. So anyway, now gene amplification can be performed by PCR, that is to be used in gene therapy. A specific gene or DNA segment is multiplied. So that's about the role of what is called, we can say, the applications of PCR, either in evolution or in genome differences identification, or we can just amplify small quantities of DNA, or in forensic science, or in gene therapy. So with that I concluded under the molecular what is called diagnosis, the various events, what we got, the PCR, polymerase chain reaction, the ELISA test, etc. Now we have to go for the next one, the fifth one, transgenic animals and the applications of biotechnology. We have to go for the fifth one, transgenic animals. We'll do it in the next class. Thank you.